Vanakkam all of you. Today we are going to see about the medical microbiology unit 2. So in this video I am just simply giving the introduction about unit 2. Okay, so unit 2 it's mainly covers about the virus. So etiology, general characters, pathogenesis, symptoms, epidemiology, laboratory diagnosis, prevention and control measures. So of the following viruses: the pox virus, HSV, herpes simplex virus, hepatitis B virus, HBV, Ebola, Marburg, dengue virus, adeno, rabies, Zika, swine, HIV, periodontitis, cru and scrappy. Okay, so before we are going to see about all the virus characteristics, we have to clear with the terminology of all these things. So okay, let's see that. First one, etiology. So etiology is the study of the causation of origin. So this is the etiology. It only determine what are the factors which have come together to form the disease or else to cause the disease. So epidemiological survey is mainly important in determining the etiology. Okay, so in epidemiological studies we can get the details about the locations and gender and exposure of chemicals or any other factors, and what are all the things which is lead to the diseased condition of the people. Okay, it may be leads to the very high prone to the disease conditions or very less. So these are the things investigated by means of epidemiology. So these only determine the etiology. That is mainly the reason for the particular disease. Okay, so causation of this particular origin. Okay, causation of the disease origin. And general characteristics means general characteristics comes under the first one, structure of virus. The viruses of the many different types. This is the simple one or complex one, enveloped or non-enveloped. So each virus possesses the some of the specific characters. So we will study about this under the general characters. And then nature of the genetic material. So genetic material is a DNA or RNA. It is present in all things. Okay, so there are two types of things are there. One is DNA, another one is RNA. So particular one of each of the virus possesses the unique genetic material. It was classified into the single-stranded DNA or double-stranded DNA, single-stranded RNA or double-stranded RNA. Moreover, the single-stranded RNA with positive mRNA strands or single-stranded RNA with negative mRNA strands. Okay, these are the classification of the genetic materials. Okay, so each virus possesses particular things. So we will see about this in this general characters. And coming to the resistant nature, particular virus is resistant to some other things or environmental exposures or chemicals or any of the disinfectants or susceptible to particular things. So these are the things we will discuss about this in general characters. And pathogenesis. The pathogenesis is the biological mechanisms which leads to the diseased state. That is called pathogenesis. So by means of this we can understand the origin and the development of the disease. So it may be acute, chronic or recurrent one. Okay, the onset of disease is very short period or acute, or chronic means lifelong or long period. So recurrent means once we have overcome that infection, after that also it has a chance to arise again. That is called recurrent. So moreover here the pathogenesis, I'll give the one of the examples. So it's related to some of the life cycle also. For example, the virus, if it enters into our body means the first it will attach to the receptors in our cells. After the attachment of the receptors, it will penetrate into the cell, and after the penetration, the outer surface, right? I am telling the general structure, right? It consists of the protein coat. It was uncoat. Okay, the genetic material is present inside that coat. So the genetic material comes out into the cytoplasm. From the cytoplasm, it was transferred into the nucleus. Nucleus where the genetic materials are present. Okay, so that virus genetic material is now combined with our host genetic material. So it synthesizes the products which required by the virus particles. So after that, the proteins are all synthesized and it forms the complete virion. Okay, so and the complete virion which causes again infect the other disease during this time. So our the host mechanism is completely blocked or else suffered with some amount of infections. So that time only the disease will arise, some of the symptoms will come to know. So that is called pathogenesis. Next one, symptoms. Okay, symptoms, that something which some of the body physically disorder was indicated by means of the symptoms. For example, viral symptoms, flu-like symptoms, gastrointestinal disturbances, some of the irritability, malaise, and rashes, and sneezing, and stuffy nose, or else swollen tonsils, and unexplained weight loss. These are the symptoms of some of the symptoms of the viral disease. This was changed from virus to virus. Okay, some other symptoms also will arise for unique of the some of the virus. We will see in detail later. Epidemiology. So epidemiology is the very good method to find the reason for health outcomes and disease in a particular populations. So epidemiology is the study of the distribution of that particular infections and what are the risk factors involved in that infections, and it is not just a disease or not a disease on a specified populations. So okay, these are the things are collected by means of epidemiology. Here I am telling the risk factors, right? Risk factors may be the determinants or only the microbes infections or else any of the toxic infections or else non-contagious infections. So these will also lead to the death of the most peoples or else any of the natural calamities. Okay, these are things are analyzed by means of the study that is called epidemiology. For example, corona is it's an epidemic disease. Okay, by means of we have analyzed all these things, it is spread by means of the virus. So it is spread to the some particular populations due to the immunological suppressions, or else they are infected with some other diseases, or else related to the age, related to the gender, whatever things, they don't have any precautions. So these are the things, and they only tell, okay, you have to take that measure, this measures. Okay, after the analyzing all these things only they have told everything. In addition to that, non-contagious one, coming to the cancer. Okay, cancer is, it's not spread from one person to another person, but it was also in a leading position, especially the colon cancer in some other countries who are eating the red meat. That is the main reason for this. Okay, why the particular colon cancer death was occurred in the particular place was analyzed by means of this studies, and they have reported that. So it is used for the control of some health problems. So epidemiology is very, very important one in finding the health problems. So after analyzing the reason only, we are able to reduce or control the some of the diseases. Okay, so next one, laboratory diagnosis. So in mainly virus laboratory diagnosis, there are lots of diagnosis out there. So first one, virus isolation. So it was done by means of cell culture, and another one, microscopy based methods. This is some or else immunofluorescence microscopy techniques, and nucleic acid based methods. This is commonly used. Nucleic acid based methods means PCR or else sequencing methods, and host antibody detection. So after the infection, so our body can respond against that foreign antigen, the antibody will produce. If the antibody is in higher amount, means that is the level of infection is so higher. So if you are suspecting some other antigen and our antibody was taken, so it was reacted with that antigen by means of the methods, means so antigen antibody interaction also detect the particular type of virus. So moreover, it's a common thing. If for the HIV virus, we will go for the ELISA test. So for each and every virus also they are having the unique test. So these are the common laboratory diagnosis for the virus. So I have given this. Okay, and prevention methods. Prevention, the vaccines. So before the risk of that anything, we'll go for the vaccines, means it will control the disease, and we have to practice the good personal hygiene and protecting ourselves from the mosquito bites. That is the important one. These are some of the preventions, and nowadays we are very much aware of the preventions, right? So washing our hands and clothes should be neat and everything. Okay, so that's also comes under the prevention. And treatment, the over-the-counter medications or else nasal decongestants and cough suppressants and pain relievers. After the finding of the which viral infections, particular viral medicines also there. So it was in with the vir. Okay, the any acyclovir. Okay, so any of the medicines which ends with the vir, it is indicates the medicines for the viral infections. And then control measures, that is my means of the immunoprophylaxis or active or passive prophylaxis. This is the prophylaxis means, okay, we have to study the reason for the infection. Okay, after the arrival we have to study the reason for the infection, what is the main mechanism and functions and how to avoid that. So particular people are infected, so we have to protect the remaining people without the infections, right? So before the infectant. Okay, so for that only immunoprophylaxis means some of the immune boosting antibody prepared materials are injected into the body for the prevention like things. And active and passive prophylaxis. Active prophylaxis means the viral administered things. Okay, viral administered, it may be the live attenuated viruses which was prepared in the form of vaccines and given to the people. And passive prophylaxis from is the antibody produced from the another host. It was considered as a prophylaxis treatment and it was transferred to the another member. Okay, that is called passive prophylaxis. The whatever the vaccines, it was similar to the vaccines. So vaccines should be given prior to the infections. After the infection, the vaccine won't be work. Okay, that time we will go for the treatment. Before the infections, we will go for the vaccines. Okay, next one, the eliminating the non-human reservoirs. Okay, non-human reservoirs, if any of the things is available, means we have practice some of the measures to eliminate such things. So eliminating the vector is the mediator of the bond, right? From one place to another place, it was transmitted by means of things. Okay, so for those things we have to analyze correctly and eliminate the particular vector without damaging the biological cycle. Okay, improving sanitation. So sanitation, okay, already sanitation is mainly important one, controlling the infections of all things. Okay, and okay, up to this I will conclude. This is the simple intro about the unit 2. Okay, in upcoming videos we will see the viruses one by one in a detailed manner. Okay, and some of the questions to think. Differentiate etiology from epidemiology. Okay, you have to think about this and you have to correlate this with now COVID-19. Okay, and reason out the connection between the etiology and epidemiology. Okay, you are also connected with some of the pandemic or epidemic disease and comment me in the comment box. Okay, so how far you will understood this concept, I will know. And prophylaxis versus prevention, of course, what is the difference between these two things, what is the combination of these things. Okay, and you have to analyze the concept in a correct manner. Okay, comment all these answers in the comment box. And if you have any other queries, and don't hesitate to mail me. This is my mail ID, sivasan921@gmail.com, and you can ping me at any time or else comment in the comment box. And thank you.